We see in the background of this title slide leaves that look orange or red or yellow. And these plants are pulling chlorophyll out of their leaves as they prepare for those leaves to drop. And so this is one of the steps in the process of leaf abscission. This process doesn't happen randomly in time. Instead, it happens during the fall. And so this is an environmental response. We will talk about that today. We'll also talk about two other environmental responses. And this is flowering time, or environmental factors that control flowering time, as well as environmental factors that control seed germination. And so our outline is simply those three topics: what controls when leaves fall, what controls flowering time, and what controls seed germination. Leaves, at least in South Carolina and points north, typically abscise in the fall. So why is this? Let's start by specifying abscise is just a fancy word for falling off. So this process happens in the fall, and this is when cold temperatures become increasingly normal and temperatures drop towards freezing. So this is important for leaves because cold temperatures slow or stop the process of photosynthesis. This is because biochemical reactions generally happen more slowly when it gets cold, and also enzymes in the cells start to lose their functions when it gets cold. Additionally, when it gets cold, it might eventually drop below freezing, and freezing temperatures can damage cells that are not specifically made to survive them. We'll talk in a couple minutes about some adaptations that help some cells survive freezing. But most leaves on deciduous trees do not have those adaptations. Additionally, environmental factors like wind can cause damage to leaves, and there can also be cumulative harm of aging that occurs throughout the growing season. So the entire time from spring through summer and into fall, there's opportunity for things like insects to damage leaves, or wind to damage leaves, or simply for aging to occur within the leaves. This means by the time those leaves have reached fall, they're getting old and they might be inefficient and worth replacing. For all of these reasons, many of our trees decide to drop all of their leaves in the fall and make a new set of leaves in the spring. The trees that do this are called deciduous trees. They drop their leaves seasonally. In a temperate area like the Carolinas, this means in the fall. There are other places in the world where this happens in response to dryness whenever the dry season comes. Now, most deciduous trees are angiosperms. Remember, angiosperms are the flowering plants. However, these terms are not synonymous. There are some deciduous trees that are conifers. And remember, conifers are gymnosperms. An example is shown over here on the right. This tree is a bald cypress, and this grows natively in South Carolina, especially in swampy environments. You can see that the leaves here are all brown, but this tree is not dying. Instead, it is simply getting ready to drop its leaves for the winter, and it will regrow new leaves in the spring, like our deciduous angiosperms. So there are deciduous angiosperms, but there are also fewer deciduous conifers. Let's contrast that with evergreens. Evergreens are trees that retain their leaves throughout the winter. In a temperate area like South Carolina, or especially in areas north of us, then those leaves will need protection from the cold to survive. One protection is having a thick cuticle. This is important because it will prevent water loss when it is below freezing or when the leaf isn't getting water from the roots because the plant has become less active or because the water in the soil is frozen. Additionally, in the cells of the leaf, the cells will make extra solutes that will act as antifreeze. So by increasing the concentration of dissolved substances in the cell, it makes the water freeze at a lower temperature. It won't freeze then at 0 degrees Celsius. It might freeze at, say, -5 or -10 degrees Celsius. This, in effect, is going to protect those cells so long as it doesn't get super cold from having their cytoplasm freeze, and they will be more likely to survive. Now let's specify that evergreen trees do not keep their leaves forever. Each individual leaf does eventually fall off. The key here is that on an evergreen, the falling off of leaves is not simultaneous, and it does not happen seasonally. So on a deciduous tree, every fall, pretty much all of the leaves will fall off. On an evergreen instead, leaves will fall off occasionally throughout the year and from year to year. So there's never a time with evergreens where the tree is completely lacking leaves. Evergreens are anything that we would think of as a traditional Christmas tree would count as an evergreen. So evergreens include most conifers. The bald cypress was an exception. And this pine tree here then is an evergreen because pine needles do not fall off seasonally. There are some angiosperms that are also evergreens. For example, this magnolia here keeps its leaves throughout the winter, even when there's snow. And so it's an angiosperm, but it is also an evergreen. We're now going to switch gears a bit and talk about steps in Vfib Decision. So leaves don't just suddenly fall off without preparation. Instead, there's a 3-step process. The first step is that nutrients are moved back from the leaf into the stem, or they can be sent down to the roots or to other organs for storage. This means that the plant will not give up valuable nutrients like phosphorus or nitrogen when it doesn't need to. Second, a protective layer will form at the end of the stem at the abscission zone, and this will protect the stem when the leaf does fall off, so there's not an opening through which infection can occur. And third, the cell walls will weaken in the abscission zone, causing the leaf to drop. So we will talk about the process of abscission and how it is hormonally controlled here. There are 2 hormones involved in leaf abscission. The first one is auxin, and the second one is ethylene. Now, auxin is made in leaves as well as other places like the stem apical meristem. And so let's just draw here, if I can pull this off, what this will look like. We might have a plant stem, like so, and then somewhere coming off of it, we will have a leaf, let's say, like so, with the petiole and then the blade. Now, abscission happens at the very base of the petiole, so somewhere down in this area. The arrow should be pointing right here. And abscission will not happen so long as there's a high supply of auxin right here. So remember that auxin, and we'll just color code auxin as being green. Auxin is being made throughout the leaf. And so all of these little spots represent auxin. That means that there's a large supply here. And this auxin will move backwards through the petiole towards the stem. So it's moving in this direction. That means that during the growing season, there will be enough auxin here to prevent abscission from happening. However, as it gets colder out and the leaf stops photosynthesizing, its supply of auxin will go down. So let's go ahead and reduce the supply of auxin inside the leaf, like so. Now the signal here is not going to be as strong, and abscission can start to happen. The second hormone involved, we said, was ethylene, and ethylene will have the general effect of encouraging abscission to happen. So depending on the amount of auxin and the amount of ethylene, it will determine whether this connection remains or whether this connection breaks. We've said that abscission happens in the fall, but we haven't said specifically what signals from the fall lead to this. So it turns out there are several different signals that lead towards abscission. The first is cold temperatures themselves. Second, increasing amounts of darkness tend to favor abscission. And this makes sense because during the fall, remember, days go from being very long towards being shorter. The longest days of the year are in summer, and the shortest days of the year are in winter. Third, damage leads to abscission, and so leaves that have incurred insect damage, for example, will abscise earlier than those that are undamaged. Fourth, stress can lead to abscission, and stress can occur for a variety of reasons, including cold or disease, or sometimes too much sun exposure to a leaf not adapted for that. We've said that the timing of leaf abscission varies with temperature. Because this is true, it means that we should be able to see different timing of leaf abscission in areas or years that have, let's say, colder temperatures compared to warmer temperatures. And we will look at this and related variables in a laboratory exercise. Specifically, we'll use a database that lets us look at things like the time that leaves first come out or the time that leaves fall off trees and correlate those with the temperature in which each observation happened. We're going to move on now and talk about controls over flowering time. There are two very general categories of controls, internal controls and external controls. So internal means inside the plant. And these are things that do not relate to the environment. They can be the size of the plant or resource levels. So for example, the amount of nutrients the plant has stored. There can also be external signals that control flowering time. And these are things in the environment, such as the temperature or the day length. And many species have both internal and external controls operating at once, and it will be combined factors that determine when exactly the plant will flower. We are going to focus mostly on day length. With regard to day length, there are three different categories that plants may fall into. The first is day neutral. So day neutral plants do not respond to day length at all. Flowering is controlled by internal signals or perhaps by temperature. Short day plants are ones that flower if days are shorter than some critical length. Conversely, long day plants are those that flower if the days are longer than some critical length. Let's look at how plants actually do this. Let's start by talking about what happens with short day plants. We said that short day plants were ones that would flower if day length is less than a certain length. However, the truth is that these plants are really measuring night length, not day length. So let's rephrase what a short day plant does in terms of night. A short day plant will flower if night is longer than a certain length. We'll look at this on the diagram. In this first diagram, we have 10 hours of day and we have 14 hours of night. Of course, the two add up to 24 hours, the number of hours in a day. And so if we had a short day plant that had a 10-hour or less threshold for daytime, really what we're saying, it has a 14 hours or longer threshold for nighttime in order to flower. And so in this case, it's getting its 14 hours or more of night, it would go ahead and flower. And we can just draw in a check. Now let's look at what happens if it gets 14 hours of daytime. That's not really important. What is important is it's getting less than its 14 hours of night. Because it does not have at least the 14 hours that it's needed, it will not flower. So we'll draw an X here. The way that we know that plants measure nighttime rather than daytime is by doing experiments where we interrupt day by shading the plant for some period of darkness, or we interrupt night by exposing the plant to some short period of light in the middle of the night. Let's start by talking about what happens when we interrupt daytime with a period of darkness. So in this case, maybe the plant gets about, let's say 6 hours of daytime, then we interrupt it with 20 minutes of dark, and then it gets almost another 8 hours of light. In this case, it's not really perceiving one long day rather. Instead, it's perceiving 2 short days. There's one short day right here, less than 10 hours. There's another short day right here, also less than 10 hours. So if the plant was really measuring day length, it would perceive the days were less than 10 hours and it would flower. However, in practice, when we do this experiment, these plants do not flower. That's because they are getting less than the 14 hours of night that they need. It looks like here maybe it's getting closer to 10 hours of night, because this day period was 14, so there must be 10 left for night. So it's getting less than the 14 it needs, and it's not going to flower. Now let's see what happens if we interrupt night with, let's say, 15 or 20 minutes of sunlight. Or in an experiment, we would use some artificial light source. So here we see it's getting a short day. It has its 10 hours or less of daytime, and so if it was measuring daytime, this would allow it to flower. But in terms of night, it's not getting 14 hours of unbroken night. Instead, it's getting some period here that might be about 8 hours long, and then it's getting a second period here that is maybe closer to 6 hours long. The key is that neither of these periods is 14 hours or longer, and so it is not perceiving a long night. Because it's not perceiving a long night, then the plant is not going to flower. So in all 3 of these lower cases, it doesn't flower, because in all 3 of them, it does not get the requisite 14 hours of darkness that is necessary. So to summarize all of that, short day plants are really long night plants, and all that matters is whether the night is as long or longer than the threshold necessary to flower. Now we'll look at what happens with long day plants. Long day plants are ones that flower if day length is longer than the certain length. But just like for short day plants, we know that they actually respond to night length. This turns out to be true for most long day plants. So let's rephrase our rule. Long day plants will flower if night is shorter than a certain length. And for an example, let's imagine a plant that requires 12 hours or less of night. And we can draw that on here, like so. Here is our 12-hour mark. And so in this first example, the plant is getting 10 hours of day. That must mean it's getting 14 hours of night, because the hours have to add up to 24. Well, 14 is not shorter than the 12 hours that is this plant's threshold. Since it's not shorter, that means this plant will not flower. In the second example, the plant is getting 14 hours of day. That must mean it's getting 10 hours of night. Now, our rule is that it will flower if night is shorter than the threshold. Remember, this hypothetical plant requires 12 hours as the cutoff. So in this case, 10 hours of night is shorter than the 12 hours. That means that yes, this plant will flower. Now let's do experiments where we break up the day by putting the plant under a box, let's say, for about 15 minutes during midday. This plant then is going to experience something like 6 hours of light, a short period of dark, and then another 8 hours of light. It turns out that this doesn't change anything. It is still getting this 10-hour period of continuous dark right here. This 10-hour period is still shorter than the 12-hour threshold, which we can draw in right here. And so just like the plant above it, whose day was not interrupted by darkness, this plant will flower. Basically, the lesson there is that it doesn't matter what happens during the light period. What matters is what happens during the dark period. Now let's look at what happens if we don't divide the day, but we divide the night by adding a few minutes of light right in the middle of it. So this plant has its 10 hours of daytime, and then let's say it gets about 8 hours of night, and then it gets a break, and then it gets about another 6 hours of night. In this scenario, it's going to flower if night is shorter than a certain length. And here it has one 8-hour night, and it has another 6-hour night. But because of this break in the middle, it is not seeing this as 14 continuous hours. It sees it as being 2 different night times. Both of these night times are shorter than the 10-hour threshold. That means that these plants would go ahead and flower. So it's breaking up the night that made the difference, not breaking up the day. And this is our evidence that really long day plants, at least many of them, are monitoring the nighttime. The overall lesson then is for both short day and long day plants, the amount of darkness is more important than the period of light. We're now going to talk about how we can apply this lesson in some holiday plants. So there are 2 plants that we tend to grow and associate with Christmas. One plant that is associated with the holiday season is poinsettias. And this is a relatively recent innovation in the last 100 or so years, but we like to put these plants out around holidays. It turns out that poinsettias are short day plants. But really, from what we just talked about, we know that means they are long night plants. And so these plants require at least 12 hours of uninterrupted darkness to get them to flower. And this has to happen well before the holiday season. So if you wanted a plant to flower in late December, you would need to treat it for a couple of weeks in early October. And so early October. And the way you would do this is by making sure that you keep the plants in complete darkness for at least 12 hours. Now it's really easy to accidentally turn on a light and mess up the whole thing. So to avoid this, an easy solution is to put a box over the plant during its dark period. Let's say put it on at 8 p.m., take it off around 8 in the morning before you head off to work or school. While we're talking about poinsettias, I thought I would mention a little bit more about their care. So first, we often call the red parts flowers, but if you look at this, you can see they're not flowers. These are really just leaves. The flowers are actually really small and aggregated onto these yellow structures down here. These red leaves then are specialized leaves associated with flowers. And there's a term that we use for this in botany. They're called bracts. Really, bracts just refer to any specialized leaf that is associated with a flower. Okay, so next, let's talk about what these plants need besides their 12 or longer hours of darkness. To be able to flower, they also need to be healthy. And getting these plants to remain healthy involves varying their water levels. So after the holidays and again in the fall, it's necessary to reduce the amount of water the plants get. You don't go down to nothing, but you water them less during these periods. Additionally, we've already talked about pruning plants, and if you never prune your poinsettia, it's going to look very leggy. So this is going to look like your control in the coleus experiment that we didn't prune, and it got long branches that didn't have a lot of thick leaves on them. So to get this bushier look, then sometime during the summer, you would need to cut this plant back about halfway back along the stems and let it regrow from there. Since we're talking about holiday plants, the other one we'll talk about is the Christmas cactus. Now, some of these are called Christmas cactuses, but many of them don't flower in late December. Instead, they might start flowering in November or a little bit later in the year. Like poinsettias, these are short day plants, and they also require at least 12 hours of continuous darkness to flower. They respond to other signals too. For example, if it's too cold, that will slow the process of getting them to flower. We will move on now and talk about seed germination. And seed germination responds to a variety of cues. Some of these cues may be internal, meaning coming from inside the seed. And these will be mechanisms basically to make sure that it's been a long enough period of time since the seed was made. This will help make sure that the seed has dispersed, it's not still attached to the parent plant, and it will give it time to disperse further away from the parent plant. Seeds can also respond to environmental cues, and there are several possible cues. One is damage to the seed coat, and we'll talk about why that could be useful for a seed to know in a minute. Additionally, seeds can respond to light, and they can respond to temperature. And here's a diagram of a seed. This is just to remind you that the seed coat is the outermost layer. We can sketch that on. It's this layer out here. So let's talk about each of these cues in more depth. We'll start by talking about damage to the seed coat. Why would this be useful? It would seem like damage to the seed would be a bad thing. But really, this is providing useful information. First, it's telling the plant, or rather, it is allowing water to enter the seed. So the seed coat can be impermeable, and once there's a nick in it, then there's a way for water to get inside. Once water gets in, then the cells that have desiccated or dried out inside can rehydrate. And water is necessary for most cellular reactions, so rehydrating will allow these cells to become more active. Additionally, the embryo needs water in order to be able to expand, and it needs its cells to expand if the embryo is to grow. And so by allowing water inside, this is going to help cells expand, and it's going to, by expanding, eventually the embryo will be able to break open the rest of the seed coat and grow outside of it. So in nature, the process of getting damage on the seed coat can happen naturally. However, if we are planting seeds, then oftentimes we're keeping seeds in an environment where they will not become damaged. That means that we have to damage the seeds ourselves. And the process of damaging seeds is called scarification, because basically we have to make scars in the seed coat to accomplish this. So scarification is manually damaging the seed coat to facilitate germination. I'm going to leave you to watch a video about this. This video is linked on Blackboard, and it's just about 3 minutes long, and it shows you somebody who has done this and what the effects are. So why is seed scarification a good signal for a seed to use to know if it's a time to germinate? Well, think about what would cause this in nature. This could happen if seeds get rubbed against rocks in the soil. Well, if a seed is being rubbed against rocks, that's really an indication that the seed is in the soil, not still attached to the parent plant, and in fact that it's probably embedded, not just sitting on the surface. So this means it could be in a good position to start growing and have its new root already in the soil. Additionally, it takes time for the seed coat to be damaged. So that means that if the seed coat has been damaged, then maybe the seed has been dispersed for long enough that it's had a reasonable chance to get further away from the parent plant and to get at least a little bit buried in the soil. A second reason that scarification is useful is that for some seeds, it's an indication that the seed has passed through the digestive tract of an animal. And this is important because if it has done so, then probably by this point the animal has defecated out the seed or released it. So by having the seed coat damaged, the seed can be confident that it has been dispersed by an animal and is now far away from the parent plant. Secondly, the seed knows that it is now in a high nutrient environment because the animal scat it is sitting in is probably filled with fertilizers that will help the new seedling to grow. It turns out that many seeds, especially those that occur in fleshy fruits that are adapted for being eaten by animals, they actually benefit and will germinate at a higher rate if they pass through the digestive system of an animal. So for example, tomatoes can germinate after being eaten and defecated. They will also germinate without this treatment, so it's not necessary. Raspberries and blackberries both also benefit from digestion. And it turns out that animals like bears eat a lot of seeds. In case you haven't seen bear poop, this is, I think, bear poop from Florida, and you can see it is just filled with seeds. So all of these seeds, having passed through the digestive tract, are now going to have a better chance of germinating. In practice, when humans are trying to grow plants, we usually don't want to eat the fruits and then defecate out the seeds because it's messy and because it's not sanitary. But there's a variety of tricks that we can use to achieve a similar effect. And so some different tricks that are used for different kinds of seeds are heating them, even for short time periods in boiling water to break down the seed coat. Some will respond to being exposed to sulfuric acids or other acids. Some will respond to short periods in dilute bleach solution. And some benefit from physical scarification, as you saw in the video of somebody scarifying garden seeds. So this all was the process of scarification. A second environmental factor seeds respond to is light. Many seeds don't, and those that do tend to be weed species. Weeds are plants that tend to be fast-growing and disperse into environments that don't have a lot of competition. So these are plants that like to get in fast, grow, and make new seeds before the plant experiences competition. So part of the strategy is having small seeds. Small seeds are better at dispersing. The problem with being a small seed is that a small seed can't store a lot of carbohydrates or oils, and so it doesn't have a lot of energy reserves. It also doesn't have a lot of protein or other nutrient reserves, and so the seed is not going to be able to grow very far before it needs to start photosynthesizing. And if it doesn't get to a point where it can photosynthesize quickly, it will die. So these seeds are sensitive to light because light is an indication that they are very near the soil surface. That means that if it does germinate, then the seedling will not have to grow very far before photosynthesis can start. The last factor that many seeds respond to is temperature. And specifically, many seeds require a cold period. This is especially true in areas from South Carolina north, where there are cold winter periods. And so when we artificially give seeds a cold period to mimic a winter time, this process is called stratification. Now this is confusing. The words scarification and stratification sound very similar. But remember, scarification is breaking the seed coat. Stratification is exposing the seeds to cold. When we do this, we typically keep seeds in a refrigerator under moist conditions. This would be something like under very slightly dampened paper towels in a Ziploc bag, or it can be done with peat moss, and that will help reduce the amount of disease that affects the seeds while they are under this condition. And so apple, for example, is one kind of seed that responds positively to cold treatment, but there are many others. The reason that this occurs in nature is that seeds don't want to germinate just before the start of winter. They wouldn't have time to establish before they had to survive under stressful conditions. It's much easier to survive as a seed that is relatively inactive over winter, and then for that seed to germinate in the spring. By monitoring temperature until there's been a cold period and it gets warm again, then the seeds can be confident that spring has arrived and it's a good time to germinate. We will close this lecture with some final thoughts. First, Vfib Decision, flowering, and seed germination all respond to environmental signals. And they respond to overlapping signals. Both light and temperature can affect all three of these processes. Second, in temperate and boreal regions, the responses are often geared toward helping the plant avoid doing something that is not going to be useful during winter. Things like germinating or making flowers at the wrong time, and it is helping them get rid of their leaves before the winter. So a lot of this has to do with avoiding a difficult cold time of year in which to grow. And finally, some garden and agricultural plants are less responsive to environmental signals than are natural plants. The reason for this is that during the process of cultivation, humans have taken over making decisions about when the plants should germinate and grow, and the plants as a result do not have to. So put another way, if humans are able to make well-informed decisions about when the environment is likely going to be favorable for growth, then there's no reason for the plant to also try to monitor the environment and make those same decisions. So most garden seeds, for example, don't require scarification or stratification, while many wild natural seeds require one or both processes.